Morning, everyone. Thank you for attending the second week of this amazing conference. I'd just like to take this opportunity to thank Heidi and Adrian and Catherine and the team for a really astounding meeting, and I hope that that will continue this week. So I'm Carol Robinson. I'm co-chairing the session this morning. We have our first speaker, who is Jamie Wilson, a lecturer in the School of Environmental Sciences at the University of Liverpool in the UK. Jamie's research involves the development and use of numerical models of ocean biology and the Earth system in order to understand interactions between marine plankton ecosystems, biogeochemistry, and the carbon cycle. And this morning, he's going to talk to us on the biogeochemical constraints on the total biogenic carbon flux into the ocean. Thank you very much, Jamie.
Morning, everyone. So thanks for that introduction, Carol, and thanks for the invitation, Adrian, to go through all of this. So I'd like to just start by acknowledging the input from the working group listed here. So we've had a number of people kind of contributing data and contributing insights and thoughts onto this. And I'd also like to thank a couple of extra contributors who shared code and some various thoughts, so Olivia and Thomas Weber. So I'd like to start off by just kind of highlighting two things. And the first of these is that so we're looking at biogeochemical constraints on what the total carbon flux into the ocean is. And so there's quite a lot of work out there that covers quite a range of different methods. So I'm going to have to be quite brief on what those methods are. So please ask questions if you're not following. I wanted to focus on the results rather than the sort of methodology for this, but it might mean hopefully you don't get too lost. And the second thing I like to highlight is that this is very preliminary work. I've been trying to pull this together in the last couple of weeks, I think, as all of the working group leaders have been doing. It's been actually quite a big change in the way of thinking after readdressing concepts I thought I knew. So what you're going to see today is kind of like a first order kind of, you can see my thought process as I'm going through getting these plots and kind of thinking about kind of what the outcomes are. So it's going to be quite in some ways a basic talk, but it's going to be kind of off the moment. So some of these plots will be produced on the plane as I came in last night, because I was just making plots and going, oh, there's more questions. I want to know this quantity. So you're getting of the moment science, essentially. So I'm going to just jump straight in. So as Adrian introduced, as part of this initiative, we want to benchmark the kind of different components of the biological pump. And I've kind of just drawn a little cartoon of what they are here. So we have the movement of carbon from the surface ocean, which is effectively interacting with the atmosphere and placing it down into the ocean interior. So we have the traditional kind of gravitational pump, which is just particles of organic carbon sinking down passively through the water column, which we've thought about for a long time. We have the migrant pump or the active pump. So we have little organisms swimming down, pooping, and interacting with that carbon flux further down in the water column. And we have the mixing pump. So this is suspended particles and molecules of organic carbon being entrained into the ocean interior via ocean circulation. So I've just drawn kind of some idealized profiles of what that kind of carbon flux into the ocean interior might look like. So this is kind of your typical Martin curve that you have. Migrant pump, we expect, would have some input at mid-depth and a shallower for the mixing pump. So the other working groups will give you a kind of overview on each of these kind of components of the pump. But this working group is focused on really the total of all of those. So we're interested in what the net carbon flux is from the surface and into the ocean interior. So just giving you a kind of a combination of those three profiles, just to kind of give you an idea of what we're after. So for eagle-eyed of you who might have noticed that on the title slide, I said the total biogenic flux. At that point, we were going to include some results from the carbonate pump, but Greet is now going to talk specifically on her own about that. So I'll push you towards her talk, And this is really talking about the total organic carbon flux. But the reason we're interested in that, for one, it's a constraint on all three of these different pump components. So you could imagine that the total flux is going to be some sum of these fluxes with plus or minus some additional processes we don't know. But it's also the link to the kind of the broader focus and the context, which is the biological pump as a carbon sequestration function. So this net carbon flux into the ocean interior is basically the key link to then how that carbon is going to be stored on the long term. So the scope, well, so the aims of the working group, and these are the same aims that we were given by Hadrian at the start, which we're going to try and get for you. So we want to aim to get a global number for the export of organic carbon, and also get export of that per region. And the regions we're using are these illustrated here in this lovely colorful plot, which Adrian essentially just drew some straight lines to approximate some biogeochemical regions. You know, so we have equatorial upwelling regions, we have a subtropical region, we have subpolar regions. So just trying to just split apart the kind of biogeochemical regions in terms of the biological characteristics and the physical characteristics. We're going to get trying to aim for a global number of export and regional export for these and then we also want to get a vertical profile of that carbon flux for a global average and for each of these regions. So the scope of this working group specifically is that we're trying to really act in that kind of constraint form. So that really, so we made a decision early on to kind of obviously include observations, that's obvious. But when it came to models, and by models I mean any quantitative treatment of the biological pump. So this could include your kind of traditional forward models, this could be an inverse model, this could be a model taking MPP and making some assumptions about that. So we wanted to really just concentrate on models that resolve all three of those components. And the caveat for that is there's only really one currently key study that has done that. Or we want to look at models that diagnose quantities. So they don't make assumptions about what those pumps are doing, but they try and diagnose the net quantity of the quantity that we're interested in. So what we're not going to be looking at are contributions from the individual pumps, because I think the whole point is that that will emerge from this whole initiative. We're not looking at quantifying anything about ocean circulation rates. We're only concentrating on the flux of carbon into the ocean, and we're not looking at the total amount of carbon being sequestered. I drew a line at the flux point. So I'm going to organize the talk around this outline. So I'm going to organize it around the four key aims, so these kind of four key outcomes. I'm going to give you a very brief overview of the methods. Like I said, they're quite varied, so it'll be very brief. Summarize some of the key caveats and the outstanding issues that we have for each of these estimates. And then I want to end on kind of posing a question about what best metrics we need to describe the biological pump, which will be setting you up for one of the breakout groups. So to start off, we're going to look at the global export of carbon. And this is where I get a little bit shaky on the ground. This is not quite my background. But so we're looking for the net total export of organic carbon from the surface. So this is the export of particulates as well as dissolved organic matter and suspended carbon. So the total carbon coming out of the surface, which should be the equivalent of the sum of those three different components. And so this is equivalent to something called new production, which is over long time scales and large scale spatial scales. This export production should be equal to the net supply of nutrients coming up from the surface, up from the deep ocean into the surface. That's what we call new production. There's also another concept which is called annual net community production. So this is the difference between net primary production and heterotrophic respiration, or we can also define it as the difference between growth gross primary production and difference between autotrophic and heterotrophic respiration. So these concepts kind of come in different ways from various different methods. We're bringing into bear some model-based estimates. So some of these are nutrient restoring models. So they basically look at the surface and they ask for what the total flux out of the surface is, trying to constrain it against the surface concentration of nutrients. So they're basically saying, what flux do I need to maintain the ocean surface look like this in a model? And there are inverse models which will take satellite estimates of, say, MPP, and they will try and constrain these processes based on comparing it against observations. So we have satellite-based estimates. That's kind of what I've just one of the same, really. And then we have some geochemical-based estimates. So these are mixed layer budgets, looking at concentrations of DIC and oxygen, and trying to account for the incoming nutrients from mixing, from advection, and trying to back out what that net export production is. And then I've also included this one sort of out there compilation that's very different, which is from Phil Boyd's paper, which is where they try to combine estimates of individual pumps. They have effectively an estimate from working from a very different standpoint. So this is a compilation of all of the studies that we got so far. And so we can kind of put quite nicely a relative number on the global export of total carbon from the surface, which is about roughly 10 petagrams carbon a year, which Adrian kind of alluded to already. But you can see there's some quite big variation, and I've split it by methods. We have the model-based methods here. They tend to center slightly higher, much more around 12 petagrams of carbon. But all of these studies are generally quite old now, sort of 10, 20 years old. And they come from generally low resolution models. So it's possible that in the advent of higher resolution models that we might see that this estimate is kind of down. I couldn't find any kind of more recent estimates from high resolution models to compare. In terms of the satellite-based estimates, so it's taking MPP and doing some calculations with it to get to the export, quite a big variation in the use of approaches and constraints that I'll come to in a minute. And the geochemical ones are kind of centering a little bit lower than 10. So these are centering around about 8 petagrams of carbon. We have this kind of systematic variation depending on method of where in that bound we fit in terms of global export. And just to recap, so each of these panels is individual studies, and then the gray areas give you a global compilation that has been. So we can go further and start splitting it into different regions. So there's only really 2 studies that I could work from at the moment. So this is really a work in progress. So this is taking a compilation of geochemical estimates from those budget-based estimates and taking inverse model-based, which is Michael Nowicki's paper, which is a satellite estimate combined with an inverse model. So both of these approaches, if you scale up to a global integral, come to actually a very similar total flux of 9.1 and 10.2 petagrams of carbon per year. But they vary quite differently in terms of where that carbon is coming from in the regions. So I've organized this by the Arctic here, so basically northern to south, so Arctic, North Pacific, North Atlantic, into equatorial, subtropics, and into the Southern Ocean. Orientate yourself there. So you can see there's some actually big error bars on this, and they don't really kind of, they come up with very different things. So I'm not really sure quite yet why that is. But it is notable that they are very different methods, and they have very different sample coverage. So the geochemical sample coverage here, you can see is very limited in certain respects. So the equatorial region, for example, is dominated by samples of the Atlantic. Subtropical region is dominated by the Pacific, and we have very little data really across the whole Southern Ocean. So we're comparing something with quite a limited resolution to basically something where we have the full resolution. that's kind of work in progress. We're going to start to try and pull in much more of the kind of satellite-based estimates to start fill out this regional and see whether this is really, whether this uncertainty is due to just having two different studies of different types, or whether there's more. So some of the caveats when looking in this. So we have to draw a line somewhere in the surface ocean to say where we're considering this export is coming from. And that's actually very inconsistently done. There are different ways of drawing that line. So we could call it 100 meters, we could call it the annual mixed layer depth, we could call it the euphotic zone. And this is on the right here, this is a study showing the differences in, if you take a model and you estimate export production at those different depth horizons, that gives you the area in which those export estimates. So up the top here is the difference between 100 meters and annual mixed layer depth. And then at the bottom here is just showing you basically the difference, spatially, zonal average, what happens to that POC estimate. So that adds an uncertainty of about plus or minus 1 mole of carbon per square meter per year. And if we just head back to that, this can actually, this is well within the differences between these studies. So it could be that this could just be a difference in definition of the export horizons. They have different definitions. Another caveat is that actually the definition of export production itself was really varied. Quite a few studies just actually approximate it to POC export and don't consider any DOC export. Some studies where they do include DOC export just factor that in as a fixed 20% of the total. So what we are planning to do now is actually go into a proper deep dive of these studies and try and pull apart those caveats and uncertainties and see if we can expand a bit more on that. So in terms of sort of open questions, this total export production is not really a common system model output. So in the CMIP exercise, common output is export production of particulate organic carbon at 100 meters. I don't think there's an output for DOC export. So we're unable to take our best models and actually compare it against this estimate. And we currently have no major compilation of total export production from floats and gliders. I'm hoping Hervé will kind of give an update on that. It was something I didn't want to go into. I think that's a compilation that deserves its own treatment about its errors and uncertainties. Next, I'm going to move on to the vertical profiles of fluxes and how we get to that. So this is kind of, so the first sort of major method to try and derive the vertical flux of carbon in the water column has been around for a long time, and this is oxygen utilization rates. So the idea for this is that you take oxygen across an isopycnal, and that represents the approximate travel of that water from the surface into the deep ocean. And as that water mass is traveling into the ocean interior, it's being, there's respiration that's removing some of the oxygen. So as you look along that isopycnal and you see the change in that oxygen, you're measuring the time change in respiration. If you combine that with an estimate of age, so how long that water mass is traveling from the surface, you can get an estimate of the rate of that oxygen change, the oxygen utilization rate. So this is done in different ways, but this is one way where you take your data across the isopycnal, you have an estimate of age, you have an estimate of the oxygen utilization, and you regress across an estimate of oxygen utilization versus time. So this gives you a kind of basin scale or large region scale estimate of the oxygen utilization rate, which should be equivalent to organic carbon respiration. Luckily, there was a new paper that came out on this, I think a month or two ago, by Olivier Sulpice, might be a mispronunciation of his name. So this was a comprehensive global analysis of oxygen utilization rates. It removed some previous biases in terms of how you approximate the oxygen utilization with terms of undersaturation. It was a big Monte Carlo simulation that propagated uncertainties in the age estimates, the uncertainties in the observations, and the definition of the depth in terms of isopycnals. And he very very thankful for him for completely sharing his code, very open. So we've managed to resample that Monte Carlo simulation onto the JETZON region that we're using for this, which broke my computer for several days. This is a huge amount. It took 2 days to run, and it kept crashing my computer and corrupting the files. Hence why the talk was put together quite rapidly. Blame this study. So the other study is the same inverse model estimate that I mentioned earlier. So this is the global prediction of Nowicki et al. So this takes net primary productivity and ocean circulation model. It has an ecosystem model and a biogeochemical model, and it tries to find the best fit of those parameters against various biogeochemical restraints like oxygen. And this resolves those 3 different components of the pump, and it's constrained against observation. And then we have a third option, which I'm going to give some special treatment to. So this is the global profile for oxygen utilization rate, and from the inverse model. And I apologize here for my error bars blowing up because this is in log space and they're negative, so they've just blown off into infinity. So for the oxygen utilization rates, what we have here is the black line is the spline fit of the average. The red envelope is a bit confusing to explain, but it's the spline fit of the 1 standard deviation of that average for all of the data estimates. So it's the average confidence of that kind of mean, if that makes sense. And the blue dots are then the 5,000 rad Monte Carlo simulation. So that gives you the kind of the bulk uncertainty simulation. So if we integrate that vertically, we get 8.37 petagrams of carbon per year, which is in the estimates. Cool. And then this is the same from the Nowicki model. And here the red envelope is just one plus or minus 1 standard deviation. So I wanted to just split apart kind of a budget in terms of depth ranges here, kind of what that 10.2 petagrams of carbon, where it's going. So you can see most of it's going into the upper ocean in terms of 500 meters. And then below 500 meters, actually, we have a very, nothing but we have it equal. It's kind of being split across the whole deep ocean. I wasn't able to get the same information for this profile because it crashed my computer on the plane. So I'll try and get that out. But so there's notable difference here is that you will notice that this is on the same, these are on the same scale. So you can notice that the mean of the oxygen utilization is a much shallower profile than we'd have for the inverse model. And this might reflect some of the caveats. So we can also go further into splitting those into regional profiles, which you can have a quick look. And I'll come back to synthesize this information in a moment. So caveats for the oxygen utilization. So this process assumes that we have proportional behavior between the oxygen tracer and the age tracers that we use. But Wolfgang Kuver has had a couple of model studies where he's got the model input, so he has the exact respiration terms in the model, and then he's then calculating oxygen utilization using the same method and comparing back to see what he gets. And what he showed is that there's this consistent underestimate of respiration in the upper ocean. So this is the earlier study. The red dots here are the OUR estimate, and the black dots showing the actual model input. You can see there's an underestimate of, in some places, up to a factor of 4 in oceans, so depths of less than 500 meters. And then this has been updated by one of his students this year in a higher resolution model for the North and South Atlantic. And you can see again, it's seeing this underestimate. Here, the blue is the OUR and the red is the model. So this process is potentially slightly has caveats in terms of the operation. And it's because of the way that, essentially, it's the way that these non-conservative tracer and conservative tracer are being acted on by mixing. So this is quite a tricky issue because that is going to be model specific. So it varies across models. And so across models, it happens in different places, depths. And so it kind of gives an unspecified caveat on this, which is a bit of an issue. But I do find at least at depth deeper than 1,000 meters that this process is more accurate. So the last estimate I wanted to get for vertical profiles is a little bit more complex. So what this estimate is from Tom Weber's paper in 2016. So he is diagnosing the flux of, in this case, phosphate, which he's scaling up to carbon, from the ocean circulation model. So he does this by taking observed phosphate distribution. He then uses a data-constrained circulation model, OSIM, to extrapolate what the preformed component of that, so what the component of the phosphate is being evected from the surface only. And if you then take that away from your observations, you get an observed regenerated phosphate field. And this is the phosphate field that's then coming from respiration. And then he uses that model then to ask, so he then uses the circulation model to say, what is the source term that I need, given the circulation field and given my observations, to maintain this regenerated phosphate field? That makes sense. So he's basically, so it's a diagnostic approach. So he's basically saying, what is the net flux to maintain this given this ocean circulation? So he does this by basically forcing the model to try and get as close as it can to this field, given some restoration timescale. So I've been running a slimline version of this. So the original analysis tried various different ocean circulation models with different versions of it. It quantified different restoring timescales, and it also quantified the uncertainty in the observations themselves. So I haven't been able to do all of that. So I've only restricted it to just differences in ocean circulation models. And I set a surface boundary to 300 meters, whereas originally Tom was setting it slightly lower because he wasn't interested in the but the analysis is reproducing the spatial patterns of the transfer efficiencies that he finds in his original paper. So some confidence that getting the same output. But there is a slight issue, which means that it's not really suitable yet for estimating the actual magnitude of that flux. And this is because this approach has no constraint on the total export production. It's just asking you at this point in the ocean, what flux do I need to maintain this concentration? It has no concept that this is all linked to nutrient uptake and nutrient supply. It's just asking what is that number in that one space. So if we just have a fixed flow field and we have a fixed set of observations, then the total flux basically comes down to the choice of that restoring timescale. So the longer timescale you have, which means you have a slightly smoother estimate, means that you have a lower export magnitude totally. Whereas if you make that a shorter timescale, it ups it. So it's a slightly difficult estimate to work with currently. But it does reproduce vertical and regional profiles. And in this case, I've just picked the longest restoring timescale, and we get a vertical integral. So this is still kind of work in progress. So to try and synthesize all of that information about the profiles, what I've done is I've fitted a power law to those profiles. And the exponent of that power law gives you some idea of the attenuation or the shape of that profile with depth. To kind of give us an estimate of whether that profile is shallower in the water column or whether it's deeper. So on the axis here, the more negative number means that we have a generally shallower curve. And if we're up here, then we have a deeper curve. So most of these fits were all generally R squared of 0.9, so very good fits. The only ones really standing out were these two estimates here that had an R square of about 0.5 or less. So these are perhaps a bit less trustworthy. So really the punchline is here, if we do the fit against the global realization curves, we get actually quite a consistent result, which comes out around about minus 1.5. And if you took the Martin curve, which is the standard thing that's been around for 30 years, which is the power law description of the POC flux, and if you take the derivative of that to get the same flux curve which we're talking about here, then you will get minus 1.8. And so the punchline is actually for all of this effort, we haven't really gone any further than the Martin curve. So actually, consistently, these three different estimates come out as you might expect if you just reproduce the carbon flux with a Martin curve. So that's, yeah, there we go. So I split it also into regions. And so you can see there's a bit more noise here and a bit more maybe structure. So the Weber ones that are perhaps in progress are maybe don't take that too much detail. But it's interesting really that actually there's not much actually that difference across the regions. So I'm not sure whether that is because we're looking at the total flux, and so maybe the DOC is screwing up some of this. I'm not sure, but so that's a kind of open question about what that is. And then just a quick note to say we could integrate vertically across those profiles to get regional export estimates. But if you do that, the evection of DOC decouples that rate locally from where it was exported. So I just did a quick estimate using my favorite model of choice, which is an Earth system model, to give you an estimate of what that error might be, so whether you underestimate it. So if you were to integrate across the water column, then you get plus or minus 20% under or overestimate of that export production rate for that region. So that's just a kind of caveat for kind of combining these two. That's where we are so far. We managed to get estimates for all the key aims. And in terms of global scale, actually, it's quite a consistent answer. And regional scales, it's perhaps not so much, but I'm not sure whether that's currently it needs more work or there's caveats. And then we have some ongoing analyses. So we're looking at direct measures of respiration. We have some people looking at chemoautotrophy, and we're also trying to think a bit more about the impact of DOC ongoing. I've got less than 5 minutes to quickly go through something for the breakout group. So I wanted to just end on this question of what metrics we actually need to best describe the biological pump in terms of carbon storage. So typically, as a community, we always focus on export production. So what I've done here is from one of my papers last year. This plot, these are CMIP 6 models. This is the export production in terms of POC, sorry, versus the amount of stored carbon they have. You can see export production doesn't really work very well as this is for pre-industrial simulations. It doesn't work very well as a predictor of carbon storage. That's kind of the export side of it. If you look at the flux of POC in the water column, then it's perhaps a slightly better predictor of carbon storage. And this makes a difference in terms of future changes. So this is the change in that carbon storage into a future simulation. And I'd just like to point out this red line here, the model that has the smallest change in carbon storage is the UK ESM model, which has the biggest export production, the lowest carbon storage. The model that has the biggest change is the IPSL model, which has relatively high export and loads of export to depth. And what I think, and so on the lower panel here is a measure of what the ocean circulation is doing during this run. You can see that it's not really that, you can't pick out a difference between models particularly. So I think here, what's happening here is that we have a biological pump that's replumbed in different ways. We have high export but shallow remineralization versus a lower export but deeper flux to the deep ocean. And it's reacting very differently to that ocean circulation. It's giving you a very different transient change. So the high flux, high export, low deep ocean model is actually responding. So I wanted to pose a question like, what metrics do we actually need to then quantify carbon storage? So coming back to Dave Siegel's paper recently, really nice framework where you can write this out as an equation at steady state. So carbon storage is the sum of the remineralization rate at any point in the water column here, multiplied by the sequestration time of the circulation, and it gives you an estimate of where that carbon is being stored. So we can rejig this around and just get that. So we can rejig this around to get and to reframe it in terms of the process we're thinking about. So we can think of this as an export flux that's being moved across the water column. So this is a now a weighted function by the circulation. So if we want to think about carbon storage in terms of these different metrics, what this really says is we need more than one metric to think about export, remineralization, and circulation. So which of those best metrics and how do we think about that? So this is one attempt for me trying to kind of compile all this together. So along the bottom, we have export production, and we have the mean sequestration time of the biological pump, which is the combination of remineralization and circulation timescales. The contours here give you how much carbon is being stored. And then these are different model simulations that I've run. So the open circles here are models with different circulations, random circulations with different mixing properties, and the dots are differences in a biological pump. So moving the export up and down and the remineralization rate up and down. You can see we have this big space where we get kind of similar carbon storage, but for very different biological pumps. So replumbed in different ways. So the question I wanted to pose really coming out of this is, what are those metrics we need? How do we visualize it and measure it so that we get a decent quantity estimate of what's happening for biological carbon sequestration? So I'd like to just say thank you, and hopefully that all made sense and wasn't too much of a dump of information on you. I'd like to just thank the working group again and the other contributors, and thank you all for your time for listening.
Brilliant. Thank you very much, Jamie. So we have a couple of minutes for questions. If you'd like to put your hand up if you have a question and introduce yourselves. And we have a couple of runners who've got microphones who will come to you. So first of all, that was very quick.
Thank you so much, Jamie. I learned a lot. I have a couple questions. One is that we are not at all talking about this week the solubility pump, correct? This is just the biological carbon pump. So just something that I try to keep in mind is that that's something that it's outside of the scope. I guess my question is, if it's 10 petagrams of carbon per year globally, that's about equal to our annual emissions, right, globally. So I guess just another thing is it worth also estimating how much of what's exported ends up, back in the atmosphere, in the surface waters, or is that kind of next steps down the road? Because about 3/4 of that must be coming back up, right? If the oceans absorb about 1/4.
Yeah, so I guess if you're thinking of it just in terms of the carbon being stored by the pumps, then effectively we just think of it as in equilibrium. So if we're assuming that it's let's just say a equilibrium pre-industrial, then the amount going down must equal the amount coming up. If you were to break that into a time scale, so I think in Dave Siegel's paper they did this a little bit. So actually that mean sequestration time of the circulation is actually a distribution. You have obviously some in evaporation, it's much quicker to get back to surface, and deep ocean is much lower. So you have some distribution like that. I think there's some work. I think there's some work with Tim DeVries about what that might mean in terms of a transient change. But then relating that but that's all in biological pump space, not thinking about the rest of the carbon cycle and not thinking about transient changes like anthropogenic or things. But then it gets a little bit more complex. I don't know how we go from there, but I think one of the following talks, we're talking a little bit about that. So you'll get some of that broader carbon context.
Yep. Hey, Clarissa Karthäuser from Woods Hole. Hi, we met online.
Yep.
I was wondering about the coast. So if we cut off at a certain depth, what is happening with the productive parts of the coastal ocean, and also with the overlaps like filaments being carried offshore?
Mm-hmm. Yeah, that's a good question. Can we add that to the list of things to do in a working group?
Sounds good.
Yeah.
Thank you. Dave?
Yeah, Jamie, just a quick comment. The work that you showed here assumes that the air-sea gas exchanges are instantaneous. If you add the fact that carbonate chemistry takes some time, it does change the amount that's sequestered. We're in the middle of stuff looking at that. It does increase inventories, and it means that you need to be considering the solubility pump as well as you do everything. So it all becomes a mess.
You're doing that in a kind of steady state. You're doing that in
a steady state model.
So then when you've got then changing emissions and changing circulation, and then it's even another step of complexity. Yeah.
I just try to do the 90%
problem right step now. By step. Yeah. I mean, I think the approach that you've used is really great. That's a way of conceptualizing actually what that is, and then really the next step is to move that into kind of a future ocean, changing ocean space.
Thank you. Time for any more questions?
Yes. Hi, Jessica Liu, NOAA GFDL. Very impressive work. I was wondering about the carbon storage problem. Where you're looking at remineralization between 500 meters to 2,000 or 1,000 to 2,000.
Yeah.
Do you have an opinion on which of the three methods that you presented would be better for estimating that those fluxes and transfers?
Yeah, okay. So if you're thinking about kind of a deep depth horizon carbon sequestration, so then the oxygen utilization is relative, at least from what we can tell from kind of comparing that method in models, it does kind of reproduce the respiration rates at 1,000 meters and below. So that's, I guess if you were thinking about, that's direct observations, and there's no assumptions going in terms of modeling circulation, etc. So that would be the most direct thing, and it does work at 1,000.
Even with very big differences in productivity.
Yeah. Okay. Yeah. Does that make? Hopefully that answers your question.
More questions?
Thank you. It was a great talk. I was just wondering, you mentioned you compared the results of your working group with those of the Martin curve. So I was wondering whether you were just based on your conclusion in terms of the mean, or you actually considered the variation as well. So all what I'm wondering is whether you actually shrink the variance through this exercise. Thank you.
Yeah, that's a good question. I mean, I was literally making these plots this week. So yeah, should we have a chat a bit more about that afterwards? I think that's right. I think that would be a good thinking about what those global profiles mean.
One more question, and then I think we can let Jamie off the hook. No, I think everyone's satisfied. Thank you so much, Jamie.
Very
Thanks
much.
a lot.